Looks like we've got a fair few people in the chat, so I'm going to make a start. Hello, everyone. Welcome to our 11th online workshop. Today, we will be presenting two talks from the same presenter, Ron Kostanje. I'm Patrick, and I'm delighted to be back after a short spell of absence while settling into my student accommodation. You can see here it's a little bit different from what I'm normally used to. I'm a patient and a volunteer with Alport UK, and I'll be hosting today's workshop. Today, we're very privileged to have not one, but three, I think, scientific moderators. We have first Francis Flinter, the Emeritus Professor of Clinical Genetics at Guy's and St Thomas's, and of course, Neil Turner, Professor of Nephrology and at the University of Edinburgh. They'll help me put the scientific questions to our speaker. We're also privileged to welcome Rachel. Rachel Lennon is the Professor of Nephrology and Consultant Paediatric Nephrologist at the University of Manchester in the UK.
Very lean mice, and they also died at an early age. And when we did the necropsy, it showed that they had a problem with their kidneys. And so it was clear to us that there was a spontaneous mutation in this colony that had something to do with kidney disease. Long story short, we identified the mutation in the COL4A4 gene, leading to a big deletion in the protein. And this probably does not allow the protomer to form and integrate in the glomerular basement membrane, right? So with like a big chunk of the COL4A4 protein being gone, it probably can't make this protomer and then integrate in the glomerular basement membrane, leading to this familiar basket weaving with Alport syndrome. Let's see. And we can see that effect of the mutation nicely in pictures from glomeruli from the mutant mouse in our lab. It can't produce COL4A4, and so here in these two images on the left, where we stain with an antibody against COL4A4 antibody with a red fluorescent flag. You can see that in the wild type mouse, you can see this nice staining of the glomerular basement membrane. And if you look at the mutant mouse, which we called bilateral wasting kidney before we knew it was the COL4A4 gene that was mutated, you can see that there's almost no red antibody staining the glomerulus. And also if you use an antibody directed to the protomer, so COL4A3, 4, and 5 together, you can see nice staining in the wild type, and in the mutant, there's almost no staining present at all. Now, the reason why we study Alport syndrome in our lab after we established this mouse model is that you would expect that people that have the same or a similar mutation all have exactly the same disease. But this is not the case. For example, in Cyprus, there's a founder mutation, and I believe somewhere around 300 people have exact same mutation in COL4A3, and they also live in a very similar environment. But despite that, they have a large variation in the age of onset and the severity. And so this suggests that there are other genes that are involved in disease, and we call these modifier genes. And the reason why modifier genes are interested is because we can use those as therapeutic targets, where it might be difficult to do something about a mutation in one of the collagen genes. It might be easier to develop a therapy to these modifier genes. And this difference in disease severity is also something that we see in our mice. When we have our COL4A4 mutation that I just showed you in the beginning, and we move those to different genetic backgrounds in mice, we can see that and these mice also live in the same environment. We see that they have a difference in disease severity. So for instance, we see that the mice from the DBA2 strain, these mice here, they have more albumin in their urine than mice from the 129 strain. And the progression of the damage is also faster. And since the environment can't be the cause for these differences, because these mice are in the same cage, they get the same diet, it can really the only explanation is that there are modifier genes involved in the differences. And so to find these modifier genes, we need to create genetic diversity. Similar to these people on Cyprus that all have the same mutation, but each a different genetic background, we need to create a population of mice that all have the same mutation, but each have a different genetic background. And this is something that we've created here at the Jackson Lab. A number of years ago, what we did was we took 8 inbred strains of mice, they're here on the top. And the definition of an inbred strain is that every single mouse of a particular inbred strain is genetically identical. So if, for instance, here this what's called C57 Black 6J, also known as B6, which is the most commonly used inbred strain in the world. Pretty much every lab uses B6 mice. All the mice within, all B6 mice are all genetically identical. And so what we did was we took 8 different inbred strains that are genetically different from each other. And we basically mixed them up. So we crossed them randomly to create what's called the diversity outbred. And in these diversity outbred, now each mouse has a unique combination of these 8 different founder strains. And so it is very similar to a human population. And so what we did then was we took a COL4A5 mutant mouse. So this is a mouse that can no longer produce COL4A5. And these were females, and we crossed these to 100 different of these diversity outbred males. And from each mating, we then took 1 heterozygous mutant female and 1 hemizygous mutant male. And so these animals, they go on and develop Alport syndrome. We measured their albumin in the urine at 6, 10, and 15 weeks of age. And we measured kidney function at 14 weeks of age. And you can see here in the males, for instance, that you can see as a group, you can see that albuminuria is progressing, but you can also see that there's a lot of variation among the males, just like what you see in a human population. And that's exactly what we're after. Then what we did was we genotyped these 200 animals. So we determined their genotypes across the genome and then did a statistical analysis to see which regions of the mouse genome are associated with the albumin levels. In other words, we're asking the question, which parts of the genome are different between mice that have high albumin and mice that have low albumin? And as you can see here, we identified 2 genes, DGKE and PIK3R1, that were associated with differences among these mice. And so that suggests that these 2 genes are playing a role in the severity of the Alport syndrome. Now, these 2 genes are very interesting for a few reasons. First, they have recently been associated with other forms of kidney disease in humans. And this is important to us because it confirms that we can find human relevant genes in mouse studies, and that by looking at Alport syndrome, we find genes that play a role in other forms of kidney disease. So these genes that modify Alport syndrome are not exclusive to Alport syndrome. And if we develop therapies based on these genes, they may also be effective in multiple forms of kidney disease. The other advantage is that pharmaceutical companies are then usually more interested in developing drugs that would work for multiple diseases instead of a single disease. And the other interesting finding is that these 2 genes are in the same pathway, and that this pathway can modify the cytoskeleton. So these 2 genes, they both are enzymes in this pathway, and a decrease in their function would lead to a buildup of this molecule here that we know can cause cytoskeletal rearrangements. And cytoskeletal rearrangements are really important in kidney disease because the cytoskeleton is very important to maintain the shape of what are called the podocyte foot processes that are on the glomerular basement membrane. And when this rearranges, that's when you get this, what's called podocyte effacement, where you get these flattening of these podocyte foot processes, and later the detachment of the podocyte foot processes on the glomerular basement membrane. But in addition to these two genes that, as I said, were already identified as being associated with kidney disease, and other groups are already working on those, we also found this other gene that we see associated both at 6 weeks of age and 10 weeks of age. And this is a gene called Fmn1 that's on chromosome 2. And Fmn1 encodes for a protein called formin 1. And this protein binds to the actin cytoskeleton. And as I mentioned, this is really important in disease progression. So it's really interesting to find another gene that's involved in the actin cytoskeleton. And we think it drives what are called protrusions. So another group did experiments in cells in a dish with this gene and see that it can form these protrusions, it's kind of like tentacles out of these cells. And Rachel Lennon showed that these protrusions are present in human Alport patient and in Alport mice, and not in healthy humans and mice. And these are some pictures from one of her papers. Now, when we look closer at our genetic analysis, we see that the mice from our cohort that are relatively healthy have a difference in their DNA sequence that leads to an amino acid change in the Fmn1 protein compared to mice with kidney damage. So this line here are the amino acids. This is just part of the Fmn1 protein amino acid sequence. And you can see that here where the red arrow is, that the animals that have high albuminuria have this E, where the mice in our cohort that had very low albuminuria have a different amino acid. And the other interesting thing is that when we compare the amino acid sequence from the mouse with the amino acid sequence from the human, you can see that it's in a conserved region. So where the human protein and the mouse protein are very similar. And you can see that in the humans, they have the same amino acid as the mice that are susceptible to the kidney phenotype. We did predictions. So this software that can predict what happens if you change an amino acid. And the prediction software told us that this amino acid change is likely deleterious to the protein. In other words, it seems that the reason why these mice have less kidney damage is because their Fmn1 protein doesn't work. And in order to test this theory, we wanted to look at what would happen in mice with Alport syndrome if we deleted the Fmn1 gene. We would predict that they have less kidney damage. Now, fortunately for us, another investigator in Boston had already made a mouse that no longer can make Fmn1. They took out part of the gene, and so this mouse can no longer produce Fmn1. And this investigator, a number of years ago, had donated embryos to the Jackson lab, and those were cryopreserved here. And so we took those embryos and we what's called re-derived these mice. And so we got the mice on the shelf. And then what we did, so these were the mice that have this Fmn1, what's called a knockout. So they no longer can produce Fmn1. We crossed these with mice that just have a perfectly working Fmn1, but had the COL4A5 mutation. And then their offspring have, some of the offspring have the COL4A5 mutation and a perfectly working Fmn1, and others have the COL4A5 mutation and have a non-functioning FMN1. And then we measured albuminuria in these mice and compared the ones that have FMN1 versus the ones that do not have FMN1. And as we predicted, we see that indeed the mice that do not have FMN1 have a significantly lower albuminuria in the urine. We also predicted that there would be a decrease in these protrusions, and this is something that we're working on at the moment. So this is some preliminary data, but the mice that we looked at so far, we see indeed that they have fewer protrusions. So this is an electron microscopy picture of an FMN wild type. So they have the COL4A5 mutation, but they have a functioning FMN1, and we see some protrusions here. And we see the COL4A5 mice that do not have FMN1 anymore, they have fewer protrusions. And also we see that the length of the protrusions are smaller in the knockout mice compared to the wild type mice. And so with support from the Alport Syndrome Foundation, what we're now working on is a study in which we use that knowledge and treat mice with Alport syndrome with a compound called BIS-T23. And this compound can stabilize the actin cytoskeleton and hopefully mimic what happens in mice that have the FMN1 mutation. And so this is really what we're currently doing. We have mice on the shelf that we're now treating for a few days with this compound, and then we follow the mice afterwards for a number of weeks and measure glomerular filtration rate, so kidney function, and albuminuria. So where are we going from here? So we have submitted this work to the Journal of the American Society of Nephrology. And after the review, the reviewers had a number of comments and suggestions that we are addressing at the moment. One of these is to look at what is called mesangial matrix expansion. So in the glomerulus, what you see in patients is this expansion of what's called mesangial matrix. And we have developed the techniques to really nicely quantify this. And that's what we're doing for all 200 animals in the cohort, and then do a genetic analysis and see if we can find genes that are associated with mesangial matrix expansion. We're also measuring this new biomarker called NBL1, which has just been published by Andrzej Królewski at the Joslin Diabetes Center, who shows that it's correlated with end-stage renal disease and diabetic nephropathy. And we see that it also correlates with albuminuria in Alport mice. So we're measuring plasma NBL1 in all our mice and see if that indeed can be a predictive marker for Alport syndrome progression. And we're also seeking funding to expand our study. With only 200 mice, we were able to identify a number of candidate genes. But as you can see, if we go back to our statistical analysis, you can see that there were other peaks that do not have statistical evidence yet, but increasing the size of the cohort will likely provide us with additional modifier genes that we would like to follow up. So that's hopefully in the near future. Just want to acknowledge the people in my lab. So this is the current crew that are working hard and making all this possible. I want to acknowledge Yuka Takamon, who did most of the work that I presented just now. She moved on and is now a graduate student in Vancouver. And I want to acknowledge our collaborators, Rachel, Jeff, and Konstantinos, that are also involved in this project. Thank you.
Thank you, Ron. Just looking at that final presentation, I can see a lot of famous names, as well as support from a charity, which I think is, really good evidence that helping charities does support scientists and help support future research. So I guess thank you to everyone that has donated both to ASF and Alport UK so far. I've got a number of questions from the Q&A. If anyone has anything else they'd like to send in, please just use the Q&A now and we'll try and get to it. Something that I thought was really interesting, that which you very, very briefly touched on, was this idea of modifier genes as a potential target for therapies. I was wondering whether you might be able to expand on that a little bit.
Sure. So kidney disease is a very complex genetic disease.
Mm-hmm.
And so in case of Alport syndrome, it is caused by this mutation in a single gene often. But then like the progression itself depends on a lot of other genes that are playing a role in this progression, or playing a role in resistance to the progression of the disease. And every person is going to have genetic variation in those genes that are playing a role in this. And so that's really the ones that we're after. As I mentioned previously, we know it's very hard to, if there's a mutation, for instance, COL4A3, to be able to fix that mutation, or to develop a therapy to kind of like nullify that problem. Maybe finding these genes that are playing a role in also a role in the disease allows us to develop therapies that are going to be easier. So for instance, those two genes that I mentioned, those two are enzymes. And so it might be easier to develop something that can fix the problems in these enzymes, or fix the problem in the pathway. So I mentioned that dysfunction of these two enzymes leads to this buildup of this metabolite. And so maybe by taking away that metabolite in some way would allow us to at least slow down disease progression.
It was interesting to me because I think a lot of patients might view Alport syndrome as one issue, but it's interesting that a fair few genes seem to contribute to this. It's not just one thing.
Right.
I'm sure this is a stupid question as well, but would that follow that families will pass on similar modifier genes? So if my mother, for example, carried one modifier gene, I would then have a similar type of disease due to the continuation of that modifier gene.
Yes, I mean, so obviously, yeah, the kind of like the risk that you indeed, in addition to the COL4 mutation, also inherit a variant in a gene that makes you more susceptible to disease progression is definitely present, and more likely than an unrelated person.
Mm. I've got a question from the chat actually. Who wants to know why you got into Alport research? What actually drew you to this specific disease in the first place? Because there's a lot of kidney diseases and mutations that I guess you could have gone for.
Right. Well, I mean, probably several reasons. Like first, like as I mentioned, it kind of like got into it just kind of like by chance. We just found this mutation, and it turns out to be a mutation called COL4A4, and there was actually no mouse model for COL4A4 yet. And that also got me involved, kind of like in the Alport syndrome community, which is just a great community to be involved in, and a lot of nice people to collaborate with, which I think is different from other diseases often.
Yeah, throwing shade at everyone else. Yeah, I like it.
The other thing is that you can imagine that like other forms of kidney disease are sometimes more complex and are more difficult to model in a mouse. For instance, diabetic nephropathy is a lot more difficult to model in a mouse than Alport syndrome. And so also just kind of like from a practical standpoint, it is a lot easier for us to kind of like use Alport syndrome kind of like as a model disease and find these modifier genes. And then from there, look at like whether this also applies to other forms of kidney disease that are just more difficult for us to model in mice.
Okay. Well, thank you, Ron. I think it can sometimes be rare to have a presentation you follow all the way through, even if you're a patient, and it was a wonderful explanation and answers to the questions as well. I'm sure Francis has some questions because I've seen a bunch of scientific ones in the chat. Francis, would you like to take over?
Sure. That was an absolutely wonderful talk, Ron. Thank you so much.
Thank you.
I think for those of us who were involved in trying to map the gene for Alport syndrome in the 1980s, we were completely puzzled as to why there seemed to be such a variability in the way the disease affected individuals within the same families. And it just seemed impossible to imagine how we would ever be able to actually pin down modified genes. We could speculate that they might exist, but the idea of being able to identify them and study how they worked was just impossible just those few decades ago. So this is incredibly exciting. Lots of good questions here for you. First one from Neil Turner. He said, Ron, you speculated that the modified genes might have a more general role in disease progression. Have any of these genes come up in studies looking at chronic kidney disease progression in general, or in diabetes and lupus, things like that?
Yeah, so as I mentioned, so see if I can remember this correctly. So with the two, so DGKE and PIK3R1, one of them, I think it was DGKE, has been associated with diabetic nephropathy, and I forgot what the other one was associated with. And then just a few months ago, there was a study showing that FMN1 has been associated with kidney disease in a Japanese population. So it's like as I mentioned, for us, it's really important because often we get the critiques that mouse studies are not relevant to human disease or human studies. And I think we show nicely here that the genes that we find in our mouse studies have also been found in human studies. And so it shows the relevancy of our work.
FMN1 wasn't a gene I'd come across before, so I was looking it up, and one of the things it's reported to be associated with sometimes in mice is limb deformities and renal aplasia, so complete lack of development of the kidneys. Have you seen any mice with limb deformities?
So they're also like, and this gets back to these different genetic backgrounds. It really depends on which genetic background the mutation is. So if you have an FMN1 knockout on what's called the FVB inbred strain, you get these limb problems. When you move it to a 129 background, you don't see it, or it's very rare. So again, there must be modifier genes that are affecting the presence or absence of these limb problems.
Right. And somebody who's remained anonymous has asked, where does the BIS-T23 compound come from, or are you making it from scratch?
No, we unfortunately not, because it's actually pretty expensive. So no, we buy it from a chemical company, and it's this is not like a new idea. So BIS-T23 has been used in other studies for kidney disease, not Alport syndrome, but other forms of kidney disease. And from what I know is that there it was very successful. It is not a compound that could be used in humans. There's just too many side effects of it. But we know that there are companies working on kind of like derivatives of this compound that would then have fewer or no side effects.
And of course, the idea that this is potentially leading to something that could be useful in a whole range of different kidney diseases is going to make it much more attractive to pharma, isn't it?
Yep, exactly.
Francisca Lauseca has asked, have you detected any changes in the glomerular basement membrane in the COL4A5 formin knockout mice? I think she's wondering whether the protrusions might play a role in glomerular basement membrane remodeling.
Right, so that's something that we're actually at the moment, I'm looking at Rachel, working on. So we have most of the EM pictures, we just need to do the measurements.
Rachel, do you want to comment on that?
Absolutely, yeah, absolutely. So it's a great project to be part of with Ron, and we have got the data. It takes a while to get all of this through, and particularly we've been a little bit struck with lockdown, but we've got the data ready, as Ron says, to do some measurements, and will be fascinating to see. I think one of the challenges is these are, these processes, when you see them in an abnormal basement membrane, it's really hard to track them carefully. So I think that's one of the challenges that we've got. Can I jump in with a quick question to Ron on, so the form FMN1 and the other, Col4A3 and 4 are all on chromosome 2. Is that coincidence, or do you think there's potential? I know your model was Col4A5 for this experiment, but is there some linked regulation there?
Not that I'm aware of. No, it's probably just coincidence. I mean, we could look at whether indeed, like, we have all the expression data, so we could look at whether it's some co-expression and whether it's regulated similarly, but my guess would be that it's just a coincidence.
Okay, there's a question from Louise Hopkinson here. Do you have any data on the composition of the mesangial matrix in the expanded disease scenario?
We have not. We could. I mean, we definitely have the material for it to look at it. If people are interested in doing it, then they're more than welcome. We'll be happy to provide whatever is needed. It's more like it's not wanting to, but it's more kind of like just the budget and resources that we have. But it's definitely an interesting question and certainly possible to do.
Perhaps just one more question here from Richard Naylor. The formin variants in your Alport mouse background look to be hypomorphic. Is the sequence variant in a particular protein domain that has links to actin cytoskeletal interactions?
That's a good question, and it's something that we need to look better at. We haven't done that yet, but we definitely should.
Rachel or Neil, did you have any other questions that you wanted to ask?
Ron, there was just one, and it's quite technical, but it's Rachel may have an answer for this as well, and it's about the protrusions observation. So in vitro, you see that the formin mutation seems to affect the formation of protrusions, but the problem is that protrusions are also probably a general marker of severity of the injury. And I was wondering how you're going to separate a specific effect on protrusions from it's just that it's some other mechanism making the disease less severe, and therefore there aren't so many protrusions.
Yeah, I mean, at the moment, I wouldn't know how to make that distinction. Yeah, so our conclusion would just be correlative, right? And not like, yeah, so we would not be able to say like, oh, it's because of the protrusions. That's the okay.
I think what would be fascinating to work out about these protrusions is what, perhaps it's formed indirectly, but what are the features of these protrusions in terms of how they're driven? I guess that would then help us to potentially manipulate those processes, so we could figure out more directly the role that they may be having.
Yeah. So all you need to do, Rachel, is grow healthy podocytes with no mutations on a basement membrane that's full of holes.
You make it sound so easy.
Really?
I think it's easier for Rachel than most people.
This is so interesting. I'm sure we could carry on discussing it all evening, but Ron's got another talk to give. Patrick, do you want us to move on?
I've been waiting all evening for the black bear discussion, so I think we really should. Ron, would you mind sharing your screen and we can get started with this this second talk?
Absolutely. All right, let me.
I think we can see it fine. We're just waiting for it to be maximized. Cool, fantastic.
All right, so as some of you British people have sometimes say, and now for something completely different. So even in healthy kidneys, we see a decrease in kidney function with age, and at some point it reaches the threshold of chronic kidney disease and even end-stage renal disease. And we know that things like hypertension and diabetes are speeding up this process, making the curve steeper. And as co-director of our aging center, I'm trying to learn what changes are happening in the kidney during aging that makes their function decrease and makes them more susceptible to disease. During COVID, flattening of the curve has become a popular saying, and it is also what we try to do. But it would be even better if we could have our kidneys recover. But unfortunately, most evidence suggests that human kidneys are unable to recover once damaged. However, there are mammals that seem to be able to do this. And here in Maine, one of these mammals actually lives just around the corner or under your bird feeder. So black bears hibernate here in Maine up to 7 months. And unlike other hibernating mammals, they keep their body temperature almost to normal. They're still metabolically active. In fact, females gave birth and nurse their cubs during hibernation. At the same time, their kidney function drops dramatically, and the literature describes the presence of kidney damage. And at the end of hibernation, a bear is basically a dialysis patient. So it has very low kidney function and has some kidney damage. But somehow it can completely recover from this, and it does this every year. And so this paper by Peter Stenwinkel a few years ago gave me the idea to try to find out how they can do this. The first possibility is that they have special genes that make proteins that we do not have. So just to make sure we're on the same page, right? So our genes are encoded in our DNA and our genome, and then when our cell needs a particular protein, it's first being transcribed into messenger RNA, and then this messenger RNA then goes to the little protein factories in our cells, and where it's translated into the protein. And so one idea would be that bears have genes, specific genes that we humans do not have. So, and to explore this possibility, we had to sequence the complete black bear genome. The human and mouse genomes have been sequenced about 20 years ago, and you can find those in databases. You can just go online and just basically grab the whole genome sequence of the human or the mouse. But this was not the case for the black bear, so we had to do this ourselves. So we isolated the DNA from a single male Maine black bear. We cut the DNA in little pieces, then determined the base pair letters of more than the 2 billion pieces, and then puzzled these more than 2 billion pieces together to get the complete genome sequence of roughly 2.6 billion letters. This sequence is now publicly available on the internet, so you can go to a database called Ensemble, and then you can just pull it up and you can see our what's called a genome assembly, and you can just look at the genes. After comparing the sequence with the human and mouse genome and some other genomes from other species, we can conclude that they do not have any special genes. And so that it must be something in the regulation of the genes. In other words, we humans have the genes that would allow kidney recovery, but they just don't get turned on or off when we need it. And of course, this is really interesting because this has the potential to then develop therapies to change this and to kind of like develop therapies that mimic what's happening in the black bear. And so to find these genes, I decided I wanted to compare the gene expression in bear kidneys before and after hibernation and see which genes were changing during this time. To get the samples, we teamed up with the Penobscot Nation, which is a tribe of Native Americans here in Maine. And they have been incredibly supportive since the beginning of our project. They allow both a spring and a fall hunt on their land, and hunters need to be accompanied by a guide from the tribe. And so with the help of Ronnie Bear here on the left of the slide, we have been collecting mostly from two of their tribal lands in Madagamon and Grindstone, which is in the north of Maine, and it's about 3 to 4 hours drive from here, from the Jackson lab. So what I do is I bring my mobile lab, which is just some stuff in the back of my car, and follow the hunters and wait until a hunter shoots a bear, and then I move in, and then I take the kidneys and process the samples in the field, and then bring them back to the lab. We then isolate the RNA from the kidneys, determine the sequence of all the RNA molecules, and then identify to which gene each sequence belongs. And then we can count how many RNA molecules come from each gene, and that will give you the gene expression of each gene. And then we compare between the two time points, and this is what's shown here in what's called a volcano plot. So each dot is a gene, and on the left side are the genes that are more expressed in the fall compared to the spring. And on the right are the genes that are more expressed in the spring compared to the fall. And then red and green means that they are statistically significant. So there are about 166 red and green dots. And the question now is, which ones are relevant to kidney recovery and which ones are just bystanders, just kind of like by accident or because they were involved in a process that has nothing to do with kidney recovery. There's no way we can test each of these 166 genes individually. It's just too much work and would be just too expensive to do. And so we needed another trick to narrow down the list. And the trick we use is to look at another species that does the same thing as black bears. And so this is what's called a tenrec, and it's a hedgehog-like animal that lives in Madagascar. And when the soil temperature drops below 20 degrees Celsius, they hibernate and they shut down their kidneys completely. So on the right-hand side is we measured kidney function using a little device. The way it works is that you put the device on the skin of the animal, then you inject fluorescent labeled sinistrin, and in a normal working kidney, the sinistrin is being cleared by the kidney, and so the fluorescence in the skin will decrease. So this device measures the fluorescence of the skin over time. You can see on the left part of this graph where they are hibernating. You can see that there's no clearance. So the line here, let's see, where am I? This is completely flat, meaning there's absolutely no kidney function. And then when you wake up the animal, you can see that the kidney starts up again, and the fluorescence is being cleared by the kidney. And so what we did was, so fortunately for us, a colleague of mine, Frank from Breukelen, who actually was a former Jackson Lab summer student, and he's now a professor at the University of Nevada. And he has a colony of these tenrecs. And he gave us kidneys collected before and after hibernation. And so we did the exact same trick as we did with the black bears. And again, here you have a volcano plot. And on the right-hand side are the genes that are upregulated after hibernation, and on the left are the ones that are downregulated or expressed less after hibernation. And then we took our data that we got from our bears, and we just compared these two data sets. And we found that there's one gene that they have in common and is significantly increased in expression after hibernation. And this gene is called TDRD5. Now, this gene is part of what is called the PIWI pathway. So I talked about how DNA gets transcribed into RNA and then translated into a protein. Well, there are several families of very small RNA molecules that can bind to the RNA molecule. So this is messenger RNA molecule that normally gets translated in a protein. there are small RNA molecules that can bind to the messenger RNA, and that prevents it from being translated. And so in this way, you can regulate protein expression. Now, this TDRD5 is involved in the production of these small RNA molecules called piRNAs. And so what we think is that with the increased expression of TDRD5 after hibernation, you get more of these piRNAs that then are able to turn off genes, preventing the expression of proteins that would normally have a negative effect on the kidney. Now, TDRD5 has only been described as being expressed in the testes. And so our first question was, where is it expressed in the kidney? And so we obtained an antibody against this protein from someone who is studying it in testes, and we used this on the bear kidney. We labeled the antibody in a red fluorescent molecule, so we can see it under the microscope. And we see that it really nicely, so this is a glomerulus, and we see that it really nicely stains the outside of the glomerulus. And we think, although we still have to do a co-staining to be absolutely sure, but we are pretty sure it's what's called the parietal cells of the glomerulus. And so this is a cartoon of the glomerulus, and these are the parietal cells on the outside of the glomerulus. And the interesting thing is that with these parietal cells is that in a very young age in humans and mice, when podocytes or tubular cells get damaged, these parietal cells can differentiate into podocytes or tubular cells and replace them. And that's something that gets lost very soon after birth. And when we do staining in very young mice that still have the ability to recover from podocyte loss, we also see this TDRD5 expression in the parietal cells in these young mice. When we do the staining in older mice, so adult mice, we do not see this. And so it seems that TDRD5 coincides with the ability to regenerate. And there's some more evidence that TDRD5 could be involved in regeneration, especially in lower organisms. So on the left is a tunicate, which is a kind of sponge, lives in the ocean. And the tunicate is an animal that can do what's called whole body regeneration. You can just cut off a little piece, and then it is able to completely form a complete organism again. Now, when you inhibit the pathway in which TDRD5 plays a role, then the tunicate is no longer able to do this. So it's no longer able to regenerate. In C. elegans, which is a little worm that's often used in the lab, it's a model organism, it can regenerate its axons. But when you inhibit the pathway, it's not able to regenerate its axons. And so we're now seeking funding to dive into this pathway and to understand how it is involved in regeneration, and if we could turn it on in the mouse to restore kidney function. So we're currently developing a mouse where we can regulate TDRD5 expression. And so the idea would be to induce kidney damage in these mice, turn on TDRD5, and see then if indeed it is able to recover from this kidney damage. So just kind of like in conclusions, bears do not have special genes for hibernation phenotypes. They have pretty much the same gene set as we humans have. We identified 160 something differentially expressed genes in the bear kidney, which are associated with hibernation. We see that there's a possible regulation of gene expression by what are called piRNAs, these small RNAs. And TDRD5 is upregulated during hibernation in both the black bear and the tenrec. And TDRD5 is expressed in parietal cells after hibernation and may be involved in regeneration and recovery. And that's something that we're currently working on. And so again, acknowledging the people in my lab that are working on this project, a number of really important collaborators, As I mentioned, the Penobscot Indian Nation, and specifically Ronnie Bear, who just had to put up with me staying in the woods for days and days to just wait for kidney samples. Ryan and Alan, who also helped us get kidney samples, and then a number of external collaborators that are helping us with this study. Thank you.
Ron, I think the sentiment from the presentation was expressed by a questioner who about halfway through asked, how the heck do you test the kidneys in black bears? And thankfully you provided an answer for that. Are there any other animals that do this? I mean, just thinking aloud, it seems like black bears and tenrecs are quite hard to domesticate, or at least not widely available. Are there other animals that might be applicable for this test?
Right.
In addition to you, of course, building a mouse model.
Right. Well, I mean, there are other, obviously there are other species that are hibernating and,
Sure.
and also dramatically decrease their kidney function.
Mm-hmm.
The thing is that these species also dramatically lower their body temperature and their metabolic function. And so I feel that those are less suitable to study, because it's just like the whole physiology is completely different from us humans. I think the bear, although of course it is difficult to get samples and to do studies, the nice thing is it's approximately the same size. The kidney is approximately the same size. When they hibernate, their body temperature really only drops 1 or 2 degrees. They remain metabolically active. And so in that respect, they're much more similar to a human. And so I think in that way, it's a better model than, for instance, like a ground squirrel that's really like its body temperature goes, that's like freezing point and has no metabolic activity.
A few people I think were wondering, obviously you've brought this up because it could maybe down the line have future application in Alport syndrome. I think a few people were curious what actually happens to the kidney that causes this breakdown that we see, that requires, I don't know if regeneration is the correct word, but what actually goes wrong in the kidney that requires it to be restored after the end of the hibernation period?
Well, I think there's a lot of, I mean, well, really the short answer is we don't really know. I mean, we have been looking at a lot of the histology of the bears to see what is happening. We do see that there's some glomerular sclerosis, and that has been published by others that looked at bear kidneys. We see detachment of tubular cells in the bears that we collected shortly after hibernation. And the interesting thing is that as we look at samples that are collected further away from hibernation, so later in the season, we see this like less and less. So it's obviously something that they recover from, this tubular detachment. But really very little research has been done. And so it is, and as I said, I mean, it's difficult to get these samples. So to get enough sample size to really study in detail what is happening is difficult, but it's something that we're working on.
Sure. It looks like quite an operation to manage to get. I understand the problems of scale. I think a lot of people were wondering if the damage was particularly similar to Alport syndrome in some way, but
no, I mean, and I think that like in relation with Alport syndrome, I think it's again, it's kind of like the underlying idea is not really specifically kind of like attack the mutation in Alport syndrome, but more kind of like the disease progression afterwards, and like can we develop therapies that would allow to slow down disease progression, or and like when like you have podocyte loss, being able to recover from podocyte loss.
Mm. Okay, well, thank you so much, Ron. Once again, really clear answers to questions and an amazing presentation. I would say I'm going to hand it over to Rachel and Neil, but I'm sure Francis might have some questions as well. But Rachel, do you want to start us off?
Sure. So many questions coming in, and I wonder if I could just kick off with one, and whether there are any longitudinal data, Ron, in the bears or these animals to indicate what the effect on baseline kidney function is of successive hibernations.
Mm-hmm. Yeah, there's not, and it's something that I would really like to do. What it would involve is to trap bears, give them a GPS collar so we can follow up on them, right? And then indeed, long term do these kind of studies. It's something I really would love to do, but
it's
has
such
not
an
been
amazing
done.
project. I can see there's so much potential. There's a question from Richard Naylor. It looks like TDRD5 is expressed in response to injury. Do you see its expression in models of glomerular injury, such as the Alport mice? You've perhaps touched on that already.
Right. It's a good question, and the answer is going to be really honestly, we haven't looked.
All right. And then a question from Charmy. Have you come across any microRNAs involved in the regulation of any genes associated with Alport syndrome?
Oh, as a matter of fact, so we're looking into that at the moment. We are also, so I haven't been looking at microRNAs in mice and in Alport mice. And I'm not sure if that's been done, but we have looked at microRNAs in the bears, and we do find a number of microRNAs that seem to target genes that are involved in kidney damage. And so that's indeed something that we're looking into at the moment.
Yeah.
So we have, like, on one hand, these PIRNAs,
Yeah.
which are slightly bigger than microRNAs, but both have a similar function, as in they are able to kind of like regulate gene expression.
Yeah. I guess the one that's relevant in terms of Alport syndrome is microRNA 21, which was found to be upregulated in fibrosis, and actually when inhibited in Alport mice.
Yeah, that's not one of the ones that we found.
No. Okay. So then from Joy, the question we might have covered already. I think we have. Oh, well, no, it's asking, do you actually take a biopsy from, so you're taking tissue and looking at histology as well in the bears? That's what you're preparing out in the field?
Yes. So far, because we don't want to, we really kind of like, we don't want to harm any additional animals. So the philosophy that we had at the moment, like so far, is that we just make use basically of the bears that are being hunted anyway in Maine, and so that's why we follow the hunters. And so when we have a sample, we indeed, like, we go in and we take a sample for gene expression, and we take a sample for histology. And so at the moment, I think we have about 80 samples so far.
Okay, and then that there's a follow-on question, I guess, that links to that. So when you're comparing your before hibernation and your after hibernation in terms of the sclerosis fibrosis, are you seeing a difference there?
So the sclerosis thing is actually has been published like, I don't know, like a while ago. But there was a group in Europe that actually looked at brown bears and see differences in sclerosis before and after hibernation.
Okay, and one from Louise Hopkinson. Have you been able to or have you looked in single cell RNA sequencing data sets to see the cell types that are producing the TDRD5?
Yes, we have. So we've looked at mouse single cell RNA-seq. and we don't. But again, as I showed, right, like in the immunohistochemistry that I showed, is that like we see a signal in very young mice, and we don't see the signal in adult mice. And so far, all single cell RNA-seq data sets are from adult mice, and so it's not something that we would expect to be there.
Sure. I'm sure Francis and Neil are bursting with questions too, so I'm going to hand over to Neil.
Yeah, what a fascinating thing, Ron. We all thought you worked on mice, and if you're going to stick with mammals, you couldn't get much more different other than by going to the blue whale. But that's just absolutely fascinating.
In fact, I have a kidney from a whale.
Wow! What, a whole kidney?
Well, it's just so we got the whole kidney initially, but it was too big to keep in the lab, so we only have a piece.
That's amazing. I used to go to a halal abattoir to get lamb kidneys for my research long ago, and yeah, quite a fascinating life hunting your own experimental resources. But like you, we were only using animals which were already going that way. Mm-hmm. So Ron, just on from the question a minute ago about TDRD5 in very young mice, have you seen it expressed in the bear kidney?
So yeah, so we so in the slides that I show you,
the one on the left was a bear.
The one on the left was a bear. Okay. Yes.
Yeah. Okay.
And so we see it in the spring, so after hibernation, and not before the spring. So
as you were talking through it, I was just thinking what human analogies we have, and I guess the best one, and it's quite a long way from it, is kidney transplantation, which interestingly has moved away from hypothermic, put them in the fridge and make them as cold as possible and hope that time moves more slowly at lower temperature. It's moved to normothermic perfusion. And I was just wondering whether, I'm sure surely someone must be looking at gene expression in the kidneys that work versus the kidneys that don't work, and comparing your results with those might just be, I mean, I've never heard of the tenrec, and actually, you know, the whole basis of this is based on that fascinating similarity. And I was just wondering whether there are other analogies which might prove revealing.
Analogies to what?
To genes that are associated with kidney recovery, essentially. So you take a kidney out of a deceased donor, mm-hmm. and you're able to keep it alive on a normothermic perfusion mm-hmm. machine, which the temperature and the amount of oxygen and the composition of the fluid has gone through lots of evolutionary stages. Mm-hmm. And I was just wondering what the gene signals are there, which are associated with recovery versus non-recovery, and whether there's any analogy to be made.
I mean, that's just an excellent idea that I hadn't thought about, but it would be indeed like very interesting to look at whether there are gene expression data sets from these kidneys, and indeed compare it to the data that we have. And as you say, like successful ones versus non-successful ones, and then mm-hmm. indeed see what the successful ones have in common that are missing in the non-successful ones. It's an excellent idea.
It's maybe not likely to be TDRD5.
Well, I mean, so
maybe it can be woken up. I don't.
Right. Well, I mean, and we focus on TDRD5 because it's the one that was in common between the bear and the tenrec. It doesn't mean that none of the other 160 something genes that we found in the bear do not have anything to do with recovery. I mean, one other one that's really interesting that maybe you saw is like SERPINC1 is one of the highest ones. Yeah. Yeah. And I believe it's known, right, that if you increase SERPINC1 in mice, that they're less susceptible to kidney damage. So that's another one that could be very interesting to follow up on.
Yeah. One of the most striking changes in these hibernating animals that seem to switch off their kidneys must be closing down their glomeruli, stopping their glomerular filtration rate. So you might expect that the target genes in this process would include things that constrict the afferent arteriole. And that was leading me to say, how much do we know about the targets of these piRNAs, which seem to be under control of this molecule?
Right. Nothing really. I mean, the piRNAs are kind of like really new on the scene, and so far people have a lot of trouble kind of like finding out what the targets of these piRNAs are. I think it's being worked out now in, for instance, C. elegans, but in mammals, I think very little is known. So it's really something that we need to start working on, and that's exactly what we're doing. So currently we're actually sequencing the piRNAs in these bear samples, which by itself is also like to isolate the piRNA is a whole like problem by itself, but we think we figured it out now. So we're really like, the next step now is to look at the differences in piRNA in these kidneys before and after hibernation. And then hopefully we can match it up with the genes that we found to be differentially expressed, right? Because it's kind of like what you would expect, that like if we see an increase of a certain piRNA and we see decrease of a number of protein coding genes, there's a good chance that they're like one targets the other. And that's what we need to figure out.
It's really difficult working with bears, and one of your concerns obviously was that you get the time points that the hunters have, and you might, if there are any very short-term changes, you know, like in the day before they go to sleep and in the day after, you'll miss them. There's a danger, isn't there, that some of what we're seeing is kind of epiphenomenon. It's part of the recovery process that was caused by something else. So I was wondering whether, I mean, you can probably do more with a tenrec.
Yeah, so I mean, so right, exactly. So, I mean, the key to like with the bears to kind of like circumvent that problem is just the number of samples that you have, right? Because like that would be true for one bear, but not for the next bear. So if you just have enough bears in each group, it would just kind of cancel each other out. It gets more noisy, and definitely the tenrec, which is in a lab where you can have animals of exactly the same age and collect the samples at exactly the same time, makes it more sensitive. And you can, like, if you can recall those volcano plots, you can actually easily see that that's the case. We found in the bear 160 something genes. In the tenrec, we found almost 600 genes, and that's just because it's more sensitive, right? So maybe there's probably more genes also different in the bear, but we just missed those because of the variation between the samples. So it's just not sensitive enough. So we're really only picking up the ones that are really different.
So are you going to do more work on the tenrec?
Yes, we are. Yeah.
Jackson Lab now has a wing full of tenrecs.
No, we stick to mice.
Okay.
Good question, Ron.
Yeah.
The thing that I find most astonishing is the huge length of time that these bears are hibernating and yet able to make a complete recovery. And I was just thinking about what happens with humans. So occasionally humans develop acute kidney injury, maybe because they're profoundly dehydrated. And with a bit of luck, with good rehydration, their kidneys will then recover. And I just wondered whether there's an opportunity there in humans to look at gene activity around the time of that acute kidney injury and in the recovery phase there, and whether that might tell us a bit about whether there are any parallel things happening in human beings. It's not really the same, because we don't tolerate acute dehydration very well at all. You've got to be recovered pretty quickly. Has anybody looked at that?
Not that I'm aware of. I think it's even more difficult to get kidneys from humans than from bears. But so to look at gene expression, and I mean, like, that brings like a whole set of other practical problems with it. But no, I mean, it's an interesting idea for sure. Yeah.
But people sometimes have a kidney biopsy in that state.
Quite late on, Francis.
Yeah.
Usually when they're not recovering, or when there's likely to be suspected to be some different mechanism for the kidney failure. On the other hand, there's lots of work on causing acute kidney injury in mice and what are the factors that lead to recovery. And actually a lot of that data is published. I haven't come across human transplant data, but there is a lot of mouse, increasing numbers of mouse data sets of gene expression. So that could be a fascinating other area to go and look at.
I suppose just occasionally a transplanted kidney is removed again, isn't it?
Usually because it's not working, but sometimes for another reason, yeah. Yeah.
I wondered, Ron, how much of this depends on that, significant drop in temperature, and whether that's what is initiating, the signals that you're seeing, and those signals having their effects on targets that we don't know about yet. It made me think, particularly in terms of neonatal brain injury or brain injury. Birth asphyxia is then cooling. Head cooling is a current treatment. Probably more challenging to cool the kidneys, because the head you can just put a cooling cap on. But I don't know. Do you have a feel for how much of this process is switched on to protect the kidneys during temperature drop?
Well, remember, I mean, so the animal itself doesn't drop its temperature, right? So it's able to maintain its body temperature, and usually finds like a place, a den, where it's protected from the temperature, from the outside temperature. So I don't think temperature is really a big issue in that sense.
We're probably getting to the end and waiting for your the next league of study in this, Ron, because it's absolutely fascinating. But Joy has asked a question which I'm just going to bend slightly. I mean, she's gone back to the transplantation question. So these bear kidneys sound like they'd be fantastic for transplantation. They're big enough to power a human, and it looks like you can put them. You're going to tell us how to put them to sleep for an indefinite period, and you can have a freezer full of these things. So there are actually a few obstacles there, like the immune challenge. We still haven't got over that, even after a lot of work on pig kidneys, for instance. There's still huge barriers there, but I think it does really illustrate the potential benefits of looking at this kind of problem, and it's a really nice thing just to reflect on at the end.
Yeah, no, I mean, and that's I think really the main reason why we picked a bear, because it's in a lot of ways very similar to humans, and so trying to figure out how they can deal with this might help us.
Tin, if that's okay, I think we're drawing to the end of our time here. Thank you so much for the discussion. I'm sure there'll be plenty of stuff that goes beyond this. I haven't seen a discussion this varied in a long time, so I think everyone's really excited about the possibility of a bear lab. I'm sure that's the next step in this research. Thank you everyone for coming today. As we continue our regular schedule of workshops into the winter, please do let us know any suggestions that you have for potential topics or research that you'd like us to feature. Our next workshop will be in a month's time. That's Tuesday, the 12th of November at 5:30 GMT. Following the recent workshops on new treatments, we hope to have a specific speaker presenting the exciting flows-in results, and of course featuring the results of a bunch of patients' organizations around the world, which we haven't done for a while. Speaking of exciting research, we saw the fruits of collaboration with the Alport Syndrome Foundation in Ron's work. We've just had a really exciting announcement come out of the States. Their 2020 research grant was awarded for a 2-year collaboration between Dr. Momita Barua, who has been featured on our workshops, and Dr. Andrew Patterson of the University of Toronto. They did a project on sex-specific genotype penetrance for predictive diagnosis in Alport syndrome. And they've taken steps to further research patients' critical questions about disease progression related to their specific sex and mutation. In other words, women are finally being prioritized in some Alport research, which is very exciting for us as a charity. Before we wrap up, we of course want to thank Ron for being incredibly patient with my questions and providing 2 very engaging talks. I think a lot of patients have come away today really excited about the research and much better informed than they were. We now know, I think, that each patient has a different set of mutations, which is really interesting and not something that many of us knew before. Thank you as well to a small army of clinicians and scientists, Francis, Neil, and Rachel, for joining the discussion. We couldn't do this without you. This workshop is built on Red Bull and extreme generosity, so thank you everyone for coming. My final thank you, and I promise this is the last one, is to Kidney Research UK for their sponsorship. They help fund our Zoom subscription and keeping the lights on at Alport UK. And this has enabled us to reach participants across Europe, the USA, and of course for the first time, Pakistan, New Zealand, Brazil, which is just so exciting for us. Now, I believe before we go, we do have a word cloud to do. So I think Archie should put something in the chat around about now. So if you would all just click on it, all you have to do is open the chat and put in one word that describes how you're feeling having seen the workshop today. Just one word, and the word cloud should slowly fill up with your comments. It's really good to see all this stuff. I think it makes presenting to you worth it. I'll pop one in myself as well, and you can guess which one was mine. Fascinated, I think, taking a pretty unbelievable, I love it. Oh my goodness, I can't believe that wasn't the title of this Zoom talk. Anyway, fantastic. Very nice. If you have any feedback about the session or you want to be added to our emailing list, email research@allport.info. That email should also be in the chat at some point as well. That's research@allport.info. Please also follow us on social media to hear updates about our workshops as they happen and details about upcoming research and in-person workshops as well once those start happening in the future. Patients can join the Alport Warriors Facebook group, and anyone can hit us up on our Twitter page. That's @AllportUK. For those of you that want a recording of this talk, we'll be popping it on YouTube and our Facebook pages later this week. Otherwise, have an amazing evening, what's left of it, and we will see you in a month's time. Goodbye.